My name is Anoop Tripathi. So I work in the plant science department. I'm starting my PhD as a Gates Cambridge scholar in plant science department in University of Cambridge. So Professor Julian Hubbard, he is going to supervise me doing my research. My research is based on one of the previous lab member name was Greg Reeves, and he discovered one method to graft monocots. So earlier it was like everybody thought only dicots can be grafted. So he came up with new idea. It's like yes, monocot can be grafted. So we published a paper in Nature this year, the starting of this year. So I'm going to continue that work and try to develop new crops. That's the whole idea. So we put like plants in major like crop plants in two. It's like a monocot and dicot. So dicot plants, what you see all around you is like a big plant, huge. They are like a dicot plants because they have, so they can, their vasculature is like in a, like a ring form. So if you graft, like you have must have see the apple tree, that big grafted apple tree. That's the dicot plant, and you can graft them very easily because they have the ring of vasculature and they have cambium. So cambium is like a meristematic tissue, and they help them to like make a vascular connection and make a really good graft. But with monocot is like maize, millet, rice, wheat, like grass family. They are monocots, and monocots the vasculature is scattered. There is no cambium, so they are really hard to graft. So the major crop we eat, they are monocot, like grass family, and we want to improve them. So it's like monocot grafting is. That's the whole point of my research. So when we start like see the plants, so my group is mainly focused on photosynthesis. So there are like two major type of photosynthesis, like C3 photosynthesis and C4 photosynthesis. So in C3 photosynthesis, there is like an extra step. It's like a toxic to the plants is photorespiration, because everything happens in like light reaction and dark reaction. Everything happen in one flow. So Rubisco is the enzyme which is fixing the carbon dioxide. They interact with carbon dioxide as well as with oxygen. So if at high temperature they react with oxygen, so they go to another pathway, is photorespiration, and they produce some toxic product, and plants had to remove them, so they waste a lot of energy. But with C4 plants, they are more efficient because they compartmentalize these two part. So they do first bit in mesophyll cells, second in the bundle sheath cell. So that's why they overcome this step, and that's why they are more efficient. So like millet are C4, and rice, they are C3 plants. So if we can introduce the C4ness of millets, like they are more efficient in photosynthesis, and they can grow in less water condition. So if we can graft them and put the more C4 character into rice, so we'll make more efficient rice. So it's like by 2050 we have to feed like 10 billion people. So we need more food production. So this is the whole idea. So like put the C4ness into C3 rice. And during my childhood, like we have summer vacation. Every time summer vacation, we go to villages. So like we study in city, go to our grandparents who lives in villages, and I was like always excited about see the agriculture thing. Like, and my grandfather, he used to tell me, if you want to like respect the farmers, you have to learn how they do the farming. And he got me interested in like how the farming works, and that thing led to me understand how complex plants are, how weather change them, how they need their nutrient and everything. And then my family is like always into science, so it's like my family either like biologists or engineers, so it's biology or math. So yeah, biology, and I had always like the biology is like really complicated. It like you try to understand it, but no, there's something left to understand. So that's what like go. And when I see the like, different kind of structure, so when I went to uni, so first day of my uni, so we had like a first practical lesson. So I made a dicot section, and dicot section was like, and it's like a double staining. I stained it and make a section, and the professor said, "Oh, that's a really good section. Everybody should come and see this." And that was like for me, it's like because you got that attention what you're like looking for, and that's the thing. I went into botany, then I did my masters in biochemistry. Yeah, that's the best thing. Like if you do some experiment, you see the result on your plant, like whatever you want, like the phenotyping of the plant, and you see is the like most rewarding thing, because you do some experiment, you plant things, and you expect I'll get something. Okay, then you plant things and wait for, and the moment you see the result, that's the most rewarding thing. And other thing, like in long run, this is going to be in the field, and for like the people you want to help, they are get benefit from it, and that's the best feeling ever.